would like to welcome all participants to the monthly UC Davis West Coast Metabolomics Center seminar series, where we invite renowned speakers from around the world, making the best use of Zooms that we learned to love in the COVID pandemic. You also see here, if you look, the people who have their camera on, Dr. Arpana Vania. So Dr. Vania will guide us through this seminar. So while the speaker will speak, please already make comments in the chat room, questions, and Dr. Vania will bundle those questions after the talk has ended, and then we will have a discussion with Dr. Tomás Barberán, if I hopefully pronounce it correct.
Very good.
As good as I can. And I see, yes, we have more people. So I just look here at the number of participants. So I repeat what I just said. The way we organize these online seminars is that you please post your questions and comments in the chat room while the speaker speaks, so that Dr. Vania can bundle those questions and we, as a starting for a discussion with our speaker at the end of the talk. My name is Oliver Fien. I'm director of the West Coast Metabolomics Center, and I welcome everyone, and especially Dr. Tomás Barberán from the CSIC. So that's, I looked that up. That is the Center of Soil Sciences and Applied Biology of Segura, of the Spanish National Research Council. Dr. Tomás Barberán has a Doctor of Pharmacy. He is one of the most cited researchers in food sciences worldwide, with an H index of 132 and 389 papers that have been cited more than 10 times. So that is very impressive. His most cited paper last year deals with elagitannins and urolithins and neuroprotection. So evidence and possible links to the microbiota, and I bet we will hear a little bit about that today, published in the Journal of Molecular Aspects of Medicine. But, he has also, highly cited papers, for example, on antioxidant activity of pomegranate juice and its relationship with phenolic composition and processing. Even that is a long time ago, still a very highly cited paper. Also flavonoids in food and their health benefits, and phenolic compounds and related enzymes as determinants of quality in fruits and vegetables. So all these are classic seminal papers that are still cited today and worth reading. His research interests are obviously, therefore, in the evaluation of the effects of different factors, both agronomic, genetic, and technology, on phenolic phytochemical compositions, and how these polyphenols can be evaluated in their activity, both in cell cultures and in animal models, including their bioavailability and metabolism in the human gut, and their effects on human gene expression. With that preface, I'm so happy to see Dr. Tomás Barberán, and welcome. The floor is yours.
Okay, thank you very much, Dr. Fien, for your very kind introduction. And I must say that I'm very happy for the opportunity that you have given to me to make this seminar in the University of California, Davis. That is a place that I love very much. I've been there 3 times, as we were commenting before. The first time was as a sabbatical stay during 1 year, and then I went 2 other times for 3 months in summer, and all the times was very profitable stay. And I'm going to focus my presentation, at least the start of my presentation on these stays in California, because you will realize what's the purpose of that. Well, I must say that I'm going to tell you a story about pomegranate and the antioxidant superpower. In 1999, we had the opportunity to go on a summer stay in California. I said we because my wife and myself, my wife is a scientist as well, And we were invited to visit Professor Adel Kader, was professor of pomology at the University of California at Wixon Hall. And then we were there working on pomegranates. We, in our area in Spain, we have a very large production of pomegranates, and in California was not really a very important horticultural product. But we were there to study the juice and the properties of the juice and the quality of the juice produced in California. Well, this is some pictures, interesting pictures from that time. In California, this is a seminar, a present seminar in the Department of Pomology. This is Professor Kader, and this is also Professor Kader with different members of the team. This is my wife, and this was my wife working on pomegranates. Why I put my wife working on pomegranates? Because this is going to be a very relevant topic that was mentioned by Dr. Finn before. One of the papers that we have produced that is more highly cited in the whole story. Well, we wanted to look at the antioxidant activity of pomegranate juice. By that time, before the end of the last century, the 20th century, there was an interest in pomegranate in antioxidant capacity evaluation, and this was evaluated in vitro, and the foods were rated according to this antioxidant capacity in vitro. So we started to work with pomegranate juice, and we were studying commercial pomegranate juice produced in California, in LA, by the company Pond Wonderful. And then we compared this antioxidant capacity with red wine, Cabernet Sauvignon, also from California, and also green tea, and juice, pomegranate juice made with arils, just with the internal part, the edible part of the juice. So there was, we were impressed for the high antioxidant capacity of the commercial juices compared to the others, but they were very good antioxidant beverages with a good reputation. And when we discovered this, the antioxidant capacity was double or triple those of the green tea and red wine. And in this study, we discovered that the antioxidant capacity was due to these phenolic compounds, to elagitannins, particularly this is punicalagin, which is characteristic of this pomegranate juice. And this is probably due to the large number of phenolic hydroxyl you have in a single molecule of this polyphenol. And then this is why they have a large antioxidant capacity. Well, this is was something very interesting. We published this in the Journal of Agriculture and Food Chemistry in year 2000. And this publication has more than 4,000 citations so far. And what was most important probably here was that after this study, the company Pond Wonderful launched this product, a very beautiful pomegranate juice that was very rich in antioxidants. And then they launched this campaign to promote the consumption of this juice, and due to the high antioxidant capacity. Antioxidant capacity was by that time related to the prevention of the oxidation processes in the body, and then oxidation was behind many health conditions related to cancer, cardiovascular diseases, etc. So this is why this was very popular by that time. This is a picture of myself in San Francisco in 2005, so 5 years later, and I was really surprised of looking at this very large advertisement in the street in San Francisco about the antioxidant superpower 5 years later that we did this study in Davis. This was also not only important for the production of the juice, but also for the cultivation of pomegranates. This is the number of acres that were available in California in 1997, less than 5,000, but there was a sustained increase in the cultivation land in California to produce pomegranates to be used for production of the juice due to this antioxidant superpower. Well, this was very important, but not only that, these are Mr. and Mrs. Resnick, that were the owners of Pom Wonderful and were the ones that funded this research on working on looking at the antioxidant capacity of pomegranates. And I was surprised to look in the web that they have given $50 million for the development of a new facility in California, in Davis. In year 2022, they presented this project that is going to be for innovation in agricultural sciences, and is going to be built by 2025, I think. And this is this couple are the ones that promoted the cultivation and the production of the pomegranate juice that was somehow introduced in the market and was very successful. Well, at the same time, there were some studies looking at the pomegranate use and cardiovascular health. There were many studies done, mainly by scientists in Israel, that they were demonstrating different studies that pomegranate use has some effect in the cardiovascular disease protection. I'm not going to say that the different activities that they described, that this was related to atherogenicity, atherosclerotic lesions, anti-hypertensive effects, anti-aggregant, etc. So, but this is something that it was associated to the phenolic compounds present in pomegranate juice and to the high antioxidant capacity of the pomegranate juice. Since then, when I went back to Murcia, to Spain, to the place where I'm working. I've been working for the last 30 years, and I started to, well, one of the objectives of our research was to look at the antioxidant activity, antioxidant superpower, and see what happened with these highly antioxidant compounds present in pomegranates once they reach the gut, and when they reach the body, and how they behave there, was the fate of these compounds. And this is was the research objective of our group from 2000 to 2012. So we wanted to know what was the fate of this antioxidant in vivo, which constituents were responsible for the effects observed in vivo by the group in Israel, and also if it was possible to produce food products with health added value out of these antioxidant compounds. So after several years of research, we discovered that the bioavailability of elagitanins, particularly punicalagin, that was the antioxidant present in pomegranate juice, was very low. So this compound reaches the gut and is not absorbed. And then there is somehow hydrolyzed. We know now that is hydrolyzed mainly by the gut microbiota to release ellagic acid, which is its molecule here. And ellagic acid is then converted by the gut microbiota in other metabolites that we gave the name of different urolithins, like this here, that they are produced from the ellagic acid by the different gut microbes that you have in your intestine. And then these urolithins are absorbed, much more absorbed than the original elagitanins that are not absorbed at all, and they circulate in plasma as glucuronides and sulfates, which are the phase 2 metabolites that normally you can see in vivo. So this means that it's not the original antioxidants, the one that are probably doing some effect on cardiovascular disease, because these are the ones that circulate and are in contact with the vascular cells. When we looked at the antioxidant capacity in vitro of the original punicalagin that is present in the juice, and also the urolithin A, that is a metabolite produced by the gut microbiota from punicalagin, at the same concentration and at two different methods of measuring the antioxidant capacity in vitro, DPPH and ABTS, we see that the antioxidant capacity was very high for the punicalagin. You can see 15 micromolar or 8.4 micromolar. But when you look at what happened with the microbial metabolite, this antioxidant capacity disappears. At the same concentration, practically you see that there's some antioxidant capacity, but it's very low compared with the original product. So then we put in question what was the antioxidant capacity of the juice. And if the real metabolites that are doing some effects in vivo are the gut microbiota metabolites. So you look at the different ligitannins, not only the pomegranate ligitannins, but also from other food products. And we discovered a different series of metabolites produced from them than the different urolithins by a sequential loss of different hydroxyls in the molecule, and to reach the final metabolites that you can see in circulation that are urolithin A and urolithin B and isourolithin A. So then the important thing is that these urolithins that are circulating are the ones that we should look for and we should study in order to see the final biological effects of the ligitannins, the original antioxidants that are present in pomegranate juice. This was started to be discovered and published in the years 2003, 2004. Since then, there's been a lot of research, and it's still going on, very, very strong research, looking at the biological effects of these urolithins, the gut microbiota metabolites of ligitannins. But mainly you can have many studies, in vitro assays with human cell lines, looking at different anti-inflammatory effects in vascular cells, in intestinal cells, in antithrombotic activity, anti-proliferative activity as well, and many other activities that I cannot show here. But also there are some studies in vivo with animal models, preclinical studies with colon inflammation model. This study we did in 2010 was very important because we discovered the anti-inflammatory effect of the urolithins. Also in Alzheimer models and muscular function models with animals, you could find that the urolithins have some relevant effects. But there is just a very few studies in humans, because they have to be done with a pharmaceutical approach, not with a nutritional approach, because you don't know really what is the exposure of these urolithins once you give the ligitannins. It's important to say that in the last few years, there's been more publications in top journals. The first one was published in Nature Medicine and was about the effect of urolithins in the muscle of rodents, particularly I think it was rats. And in this case, they discovered that it was related to mitophagy, and it was a very good publication. But now there's been other studies on neuroprotective effects of urolithins and colon inflammation diseases. Particularly, we have recently published something on this topic with in the journal Nature Communications.
No, what I'm looking for just this.
Sorry, with some colleagues in Spain and in Switzerland, and we have in this study what we did in Nature Communications. What we published in Nature Communications was that the urolithins, they can reach the brain, particularly the sulfates, and the aglycons can be there. In this study that we did in mice, and it was really very, very interesting, because it means that these compounds can have an effect beyond the intestine, and they can reach the brain. And also there's been a study in humans in which they see that they show some mitophagy as well and muscular effects, and this was somehow a very important paper, because it opened the possibility of using the urolithins as charge in nutritional supplements or drugs. Unfortunately, the number of publications that we have in vitro and in animal models is much higher than in humans, and the relevance is just the opposite. So we need to focus into more studies in humans in order to demonstrate what's the effect of these compounds, these metabolites. So what's important is that for polyphenols and health, we need to consider in order to know the effects of polyphenols in human health, gut microbiota. Gut microbiota cannot be disregarded in this kind of studies because it's responsible for many of the metabolism and the metabolic changes that are important. So the next question was, do we all have the same gut microbiota and the same kind of metabolism due to this gut microbiota in our body? Well, we know that there's large variability in the human gut microbiome. There's many publications in different journals looking at differences in the main groups of bacteria that you have in the gut. You have here the enterotypes, although they are discussed, but this is something that was already a possibility. Also, the diversity, there is a large diversity between the different individuals, depending on the number of taxonomic units that you can find in the gut. Also, there are differences in the quality of the composition of the gut microbes. If there are different bacterial groups, you see that there are different barcodes for every individual that is different. So there's different somehow the quality of the different bacteria, and also in quantity there are differences between the different individuals. So it's important that we don't have the same gut microbiota. We have to consider that after the dietary intake, we have to face the gut microbiota metabolism plus the human metabolism of the same phenolics. So we have a very important role for metabolomics, because we need to deal with this high number of metabolites that are produced by the gut microbiota and the human metabolism. And I'm going to show you now some examples about this. I'm going to talk something very briefly about the metabotypes of phytochemicals, the gradient of production of different metabolites in some volunteers, the clusters of metabolites. Particularly here, I will talk about the berries and nuts, ellagic acid, and the clusters of metabolites, the citrus metabolites, lettuce sesquiterpene lactones, saffron carotenoids, and then something about bile acid metabolism and amino acid metabolism, if we have time. I'm going to say something about phenolic phytochemicals and gut microbiota metabolism. Well, these are phenolic compounds. You know that these are present in many different food products, but in this case, we have foods that are rich in particularly a group of phenolic compounds, and all of them, they have the characteristic, in addition of being of plant origin, of course, is that they are considered somehow healthy, and they contain large amount of different types of phenolic compounds that I'm not going to describe now. What happened with these phenolics when we eat them? Well, we ingest, and then they meet the gut bacteria that metabolize these phenolics. Well, this is what happens for many of the phytochemicals, the phenolic phytochemicals. They have a large molecular weight. They are not absorbed in the small intestine. And imagine condensed tannins, ellagic tannins, self-associated ellagic acid, juice cloud compounds, like the case of flavanone rutinoside from citrus fruits. These are not absorbed, and they reach the colon, where they meet the gut microbiota and is doing a lot of biochemical conversions to release small molecules that are much better absorbed, and they circulate in plasma, and these are the ones that are producing the health effects finally. Well, these are different examples of phenolic compounds from fruits. Here is the flavonones from citrus, isoflavones from soybean, lignans that we can have in many different seeds and seed products, ellagitannins, phenylpropanoids like these ones. All these compounds are not always absorbed. Many times they reach intact the colon, where they suffer a number of metabolic reactions. The first one is hydrolysis. There's a number of bacteria that is responsible for this hydrolysis in the small intestine and also in the colon. This leads to all the glycosylations of the different compounds, of the different phenolics, and then some ester hydrolysis as well. And then there's some cleavage. This is a bit more complicated than the hydrolysis. In this case, you need the special enzymes that are produced by the anaerobic bacteria in the colon. And this is present in different members of the Egertolaceae family and other bacteria, that they are producing cleavage of the molecules and releasing this part of the molecule after ring cleavage. And you see this the compounds that are metabolites that are generated from the original compounds through the action of this bacteria. And finally, there is a reduction effect, a reduction activity that is reducing the double bonds, like hydrogenation or the hydroxylations, removing the hydroxyls as well. And this is a common feature in all the different phenolic compounds. And finally, these are the compounds that you can find in circulation, like enterolactone and enterodiol from the lignans, or the urolithins from elagitanins, or a quol from the isoflavones from soybean, or this kind of small phenolic acid, phenylacetic, phenylpropionic acids that they are derived from the fragmentation, the breakdown of the flavonoid molecules. And this is something that is important to see the complexity of what happens in vivo in the phenolic compounds. I'm going to say something about the gut microbiota metabolism. Remember what we were talking about, the punicalagin in the gut. We know now that different lactobacillus and enterococcus bacteria, they are releasing ellagic acid in the small intestine. It's not just the conditions that you have in the gastrointestinal conditions of the enzymes, human enzymes, and the pH, etc. Is this bacteria that is releasing ellagic acid. And we know now that we need at least a consortium of 3 different bacteria in order to produce the conversion of ellagic acid into the final metabolites, urolithin B and urolithin A, that are the ones that circulate in plasma. So there's 3 different genera that we have discovered: Gordonibacter, Elagibacter, and Enterocloster, that together they are doing the whole conversion. In some cases, you need 2 bacteria to produce. 2 bacteria are able to produce this, but in others, there's you need a special one in order to have the final step, whatever. So this has been already described the last year in the Journal of Agriculture and Food Chemistry, and now there's a patent that we have made on this in order to be able to produce the different individual urolithins through the use of this bacteria that we have now isolated. So this is very exciting to know. Something that is important to say here is, when looking at metabolomics, is that we can have clusters. Clusters means that we have molecules with the same structure, same formula, and the same mass, exact mass. And then you see that in this case we have tetrahydroxyurolithins, different molecules, different UV spectrum, but very similar or identical mass spectrometry behavior. We also have trihydroxyurolithins, another cluster. So far, 3 of them, but there's more of them to come. Then we have another cluster of two of them. And finally, we have just one with 1 hydroxyl. So this is what is already known. But if you look at what happened with other fruit products, we have also discovered through metabolomics that we have other new urolithin metabolites, that they belong to the same kind of clusters, but this is the R metabolites. This is urolithin M7R, urolithin AR. These have the characteristic that they have removed this hydroxylin 3 position of the ellagic acid, and then there's bacteria that they have the possibility of doing this dehydroxylase activity, and then they produce these new metabolites that they are also located in some volunteers, in addition to all the metabolites that are normally produced by the rest. So in this part of the figure, I would like to show you that we can have this kind of clusters that I mentioned. When we look at tetrahydrourolithins, you see that there's 5 different isomers that you can have a look here. And when you look at trihydroxyurolithins, you have other molecules, 10 in this case, and dihydroxy another 10. Only the blue and the red ones are the ones that have been discovered in humans so far. The others are feasible and could happen in the future, but not now. In addition to this complexity, if we look at phase 2 metabolism, we know that there is a number of conjugation with glucuronides and sulfates, and this increases the number of molecules that you can find in your metabolomic studies. The problem with these glucuronides is that when you look at the separation they have exactly the same formula and the same in the same mass, but you look at the chromatogram, 3 of these metabolites, these 4, elute together very close, very difficult to separate, and 1 of them is separated. The UV spectra of some of them are different. Isourolithin is this one, and this is urolithin. But normally in the metabolomic study, you don't use this, so it's not possible to differentiate them. But recently, we have found that with supercritical fluid chromatography, you can separate the 5 of them, but this is not very sensitive, let's say, or not accurate in separation. So it's something, there's a space for improvement in the analytical methods. Well, I'm going to say something about the phenotypes of the Lagitani microbial metabolism. Not all the volunteers produce the same, and this is what I'm going to show you now. There's a group of volunteers that they produce only as a final metabolite urolithin A, and we call this metabotype A. Also, there's another group of volunteers that in addition to urolithin A, they produce isourolithin A and B, and this is metabotype B. And another one is a group of volunteers that they are not able to produce relevant amounts of the different urolithin. Normally they are below the limit of detection, and this is metabotype 0. Well, if you see here, I gave here something that is funny that I want to show you. This is the metabotype 0 is black, metabotype A is red, and metabotype B is blue. And when you look at the logo of your metabolomic center, you see you have the same color, 3 individuals with the same colors. That probably this means that there is some differentiation in the metabotypes of these individuals, and this is I'm very happy to see that we have the same idea and focus into the colors and the way of showing these differences. Well, when you look at the inter-individual variability of the different metabotypes A, B, and 0, if you look at individuals that are healthy, the main group is the metabotype A. Metabotype B is less relevant, and 0 is not very relevant. But if you look at individuals with gut dysbiosis, like colorectal cancer, metabolic syndrome, and many others, in this case, metabotype B is getting more relevant. And also, it's important to say that this also is more relevant with age. With people, when you have older people, they have more this metabotype than the others. So this is very important because sometimes when you look at the biological effects in vivo in clinical trials with polyphenols, it's difficult to find significant results with the whole population because there are some responders, some non-responders. So it's difficult to have significant results. But probably these are significant for specific groups of, and in this case, this is the approach we did under the framework of a European project. The project Bacchus was on cardiovascular benefits from food bioactives, and in this case, this study was the PORME-CARDIO trial that we did a double-blind, 2-arm, placebo-controlled, randomized crossover dose-response trial for 6 months. And in this case, we gave an extract of pomegranate rich in punicalagin, and we gave this to close to 50 volunteers. And we measured different cardiovascular disease, cardiometabolic risk biomarkers. And one of them, this is the case of total cholesterol. And you see that there's large variation in the cholesterol level in the different volunteers, but metabotype A was somehow in a very healthy situation, but metabotype B was in a very, a more complicated situation at cardiovascular risk. But when we look at different, after the intervention, when we looked at the different cardiometabolic risk biomarkers, when we look at all the individuals, there was a trend. These are different doses and different biomarkers, but there was no significant results. But for metabotype A was exactly the same. There was no effect, no relevant effect. But for metabotype B, in this case, we did see a very interesting decrease in the cardiometabolic risk biomarkers with the supply of the pomegranate L-argitanins as a nutraceutical supplementation. And it was particularly very relevant in different biomarkers. That was very, very, very exciting. And something else that I want to show you is that metabolomics can also be used for biomarkers of processing. And in this case, we did a study looking at different orange juice produced by different technological methods. And this was pasteurization and high pressure Holter homogenization was compared with hand squeezed juice, and we gave this to humans. And after that, through UPLC-Q2FMS, we found different biomarkers that were associated with the processing. And we found that the different biomarkers, this is the metabolite that you can find in the citrus, in the orange, and this is gut microbiota metabolites or human metabolites produced by different biochemical pathways, and the flavonoids and terpenoids were somehow metabolites, and somehow there was, this is the intermediate metabolite, and this is the urine biomarker that you can find in the body that we analyzed. But what was important is that we found this metabolite that was a polymethoxyflavone sulfate that was detected and associated with the processing. And this makes sense because during the hand squeezing, these metabolites are present in the orange, in the flavido, in the orange part, in the external part. And during the hand squeezing, you normally don't put into contact the pill with the juice. But in the industrial way, these are in contact, and this is why there's an extraction of these compounds that can be considered biomarkers of processing. This is relevant because processing has now been a hot topic of research, because processing is being considered not being very healthy, particularly ultra processing, but it's interesting to find biomarkers of this, and this is why we found this metabolite that was very exciting. And in fact, we have now a study, a Spanish project funded by the Spanish Research Agency to look at the interindividual variability of the production of these gut microbiota metabolites from polymethoxyflavones, and there's a combination of CYPs that they can do this in the methylation, and there's also some gut microbiota that is able to do that. And we want to know what's the main responsible for this production of these metabolites. And well, regarding the interindividual variability in citrus flavonoids, it's important to say that we can also see this kind of situation. We can see that there is a continuous representation of the excretion, and in this case, this is like a gradient. There are volunteers that they excrete a small amount of what it is ingested, and some others they excrete a lot. So the exposure of this kind of compounds after the intake is very different depending on the individual, and this is something that we need to consider as well in the metabolomic studies. Just to continue with my talk and go to close to the end, I'm going to tell something about other constituents and the metabolism, the gut microbiota metabolism of other constituents. And in this case, I'm going to say something about carotenoids and terpenoids as the main compounds. In this case, gut microbiota can also be relevant for the health effects. First, I'm going to say something about saponins that are present in different food products, like this here that we have here on the right. And these are terpenoid glycosides that are present in the food, but they are somehow metabolized by the gut microbiota to release the A glycan, that is the one that is absorbed, and that's probably having the final effects in vivo. In the case of terpenoids, other terpenoids like limonoids, present also in citrus fruits, you see that there is a number of activities that can be done by the gut microbiota. Again, as the reductase, you can reduce this carbonyl in order to give rise to hydroxyl, and then you can have the dehydroxylase activity to release this hydroxyl and produce the metabolite. And also there is a lactonase effect, a lactonase activity that can break down this lactone to release a carboxyl, and then through the oxidation you can have a carbonyl here. So this is the kind of metabolites that you can produce with the gut microbiota in limonoids. Again, in the bile acids, it's important to say that the gut microbiota has a very important effect, converting the original bile acids into secondary bile acids through a number of different transformations, the hydroxylations, oxidations, etc., that I'm not going to comment here in detail. There's been some recent paper, this is interesting, about the microbial conjugated bile acids, that the bacteria in the gut is able to somehow produce conjugations of the bile acids with the different amino acids, and this opens new possibilities for studying and for getting some biomarkers of health, etc. We have recently in our group discovered a new group of bile acid metabolites. In this case, are the esters, in which instead the amide that is you can find in the amide in the described by the Queen group, then you can see that there is a number of esters in the hydroxyls, and these opens a new family of metabolites that are relevant because they can also be related to some physiopathological health status. What is important here is again the clusters. If you look at these are the microbially conjugated bile acids identified and confirmed in the paper what we published in Molecules, and you can see that there's a number of molecules that they have the same structure, the same mass, the same formula, and there's a number of clusters of these compounds, and this gives you an opportunity to study something very relevant through the metabolomic study. So it's important to see that this is a real challenge to know what happened with all these metabolites that give a very complicated and very complex analytical reports. Another group that I would like to mention is the terpenoids. In this case, the terpene lactones in escarole and lettuce, lactocopicrine and lactucin are these metabolites that are terpenoids. And lactocopicrine, when it is in contact with the gut microbiota through esterases and reductases, it can convert a double bond into methyl. And then the hydroxylations and double bond reductions are also feasible. So this is the kind of biochemical reaction that we saw in the phenolics, but are also occurring in the terpenoids. And also looking at lactucin, that is the hydrolysis product, reductase, and the hydroxylations are also present there. You can have a number of different double bonds that are reduced to give rise to different molecules from lactucin to reach to hexahydrolactucin. And the hydroxylation as well, you can remove the So looking at the clusters, we can see that there's a number of groups of this is the different groups of the hydrolactucins, tetrahydrolactucins, and this is the only hexahydrolactucin that you can find. And the same happens with the 8 deoxylactosine. So clusters here is again something that is common in this kind of metabolites produced by the gut microbiota from the original compound. We went and we tried to find these metabolites in vivo in humans, and we succeed to find, after the escarole intake by five healthy volunteers, that many of these metabolites were found in the volunteers, in most of them, although some interindividual variation was observed for some of the metabolites that was relevant. And the number of isomers was from 1 to 7, depending on the molecule that we could find in the urine of these volunteers. So, and I want to say something just to reach close to the end, is something that we published a few years ago, is about, this was a scientific opinion in the Journal of Agriculture and Food Chemistry, and we thought that we have to consider if the polyphenols gut microbiota metabolites are bioactive or biomarkers. This is something that we published there, and in fact, is today I can say you that this probably, or both, can be biomarkers and bioactives. Some of these metabolites, gut microbiota metabolites, can have some effects in human health, and some others can be biomarkers of the gut microbiota that we have in our gut that is related to health, or somehow is also showing us that there is some conditions for some diseases already occurring in the body. So this is something that is open, and here the bile acids, the phenolic metabolites, and the terpenoid metabolites can give us a very good opportunity to study what happens in vivo. So I want to say something about carotenoids, because this is something very new. Nobody has published the metabolism of gut by gut microbiota of the carotenoids. And in this case, we studied the pigment of saffron. Saffron is a pigment that is very important in Spain because it's used to cook the typical paella. And in this case, this is a very stable pigment. It's an apocarotenoid, and it releases quercetin after release of these sugars. And this is due to a beta-glucosidase activity that is provided also by the microbiota in the small intestine, in the colon. So you release quercetin. But now what we have learned is that in addition to this release of the egg-glycan, we have detected that there's a reduction of double bonds, of different double bonds, and also the methylation to produce a family of new metabolites that we are now writing the paper to be published. And I can tell you that this is very exciting because this can reach the brain as well and could be responsible for the health effects that are observed for or are described for the saffron, in this case, neuroprotection and antidepressant and improve mental performance, etc. And well, I don't want to say more because the time is over already. And this is about, well, the gut microbiota metabolism of amino acid is also very relevant, very important, and we need to consider as well. And due to the number of metabolites that you can reach from different amino acids. So the take-home message to end my seminar today is that metabolomics is essential in the analysis of bioactive gut microbiota metabolites. It's important that the metabolomics helps showing the interindividual variability. We know that not all the individuals are producing the same metabolites, and metabolomics is needed in order to have a whole and thorough study of these metabolites. Also, the concept of metabotypes is important to be shown because this can show different health effects after the intervention. We have already done this with the urolithin metabotypes, and I'm sure they will come later with other groups of metabotypes in humans. I would like also to mention again something about metabolite clusters that are observed in phenolics and terpenoids and are very relevant because they give you a very complex profile of metabolites. and also the human and gut microbiota co-metabolism that leads to even more complex clusters with the same mass and similar fragments. So it's something that is important to say. And just to end, I would like to say something about precision nutrition, metabolomic analysis of bile acids, amino acids, terpenoids, and phenolics can help effective integration in precision nutrition. And I think this is a very important thing that we need to continue. Well, this is just a picture of my close collaborators in Spain, in Murcia. This is Rocío García Villalba, David Beltrán, Carlos Garcia, that is working in untargeted metabolomics, and Lola, who is a technician working with us. And just thank you for your attention. This is the next picture from Spain, and I'm ready for any questions that you have. Thank you.
Thank you so much. Yes, Dr. Bonilla, would you take over?
Yes, of course. Let me just get my questions here. Thank you, Dr. Barberán. That was a great talk, very comprehensive in terms of all the information about antioxidants and how they're related to the gut microbiota. Going back to the start of your talk, the first study that you mentioned, you were measuring antioxidants in different beverages that included wine, green tea, and of course the star of the show, pomegranate juice. How is antioxidant capacity being defined or measured in that study?
Okay, well, this was, well, it's still a method that is used in many labs and for many scientists, but it's not really very reliable, because what you measure in vitro is something that does not match what happens in vivo. So, but this was a very simple method using free radicals, and these free radicals you can somehow neutralize or scavenge by the antioxidants, and this can be easily measured in vitro by DSD-PTH, APTAs. There are many other in vitro methods that can be used for detection of antioxidant capacity. But normally the antioxidant capacity also somehow parallels or correlates with the total phenolic content. If you look to phenolics, all of them, they have antioxidant capacity, and there's a similar, if you go to Follinsio-Calteo, for instance, you have a correlation with what happens with the antioxidant capacity. But the hype of this antioxidant was the previous century, in the 20th century. Now it's completely over. Nobody would like to look at the antioxidants anymore, although some companies are still using this as a market strategy, because everybody understands that the antioxidants are somehow protecting against free radicals, and free radicals are not good for your health. But the important thing is to know what happens in vivo with the antioxidants.
Yeah, that's totally true. There is a trend in the general market that the antioxidant isn't, like the big flashy thing that people are selling. It's mostly like gut microbiota and how to improve that.
Yeah.
I've definitely noticed that myself. So I think this is a great segue to kind of talk about the polyphenols in the gut and the gut microbiota that you highlighted so nicely in your talk. I think taking a step back, just to make sure we're all on the same page, are the large polyphenols mostly broken down in the large intestine, or does that already happen in the small intestine?
Well, the large polyphenols are mainly broken down in the gut, in the large intestine. The small intestine, they are not absorbed, and they can be somehow, some of them they can be hydrolyzed, but this is a minor part of the polyphenols. Most of them, they go to the colon, where they are degraded by the gut microbiota, and sometimes they are not even degraded there. They can do some local effects, but they are not degraded, and they can be excreted in feces as such.
Okay, great. And now really there's so many questions regarding ETs or elegantannins and their metabolites. So we'll take a few moments there discussing that. The first question is, are the urolithins MSMS spectra available and or the isolated compound derivatives that the gut microbes produce? So essentially, are
Yes,
the MSMS spectra available for us
yes.
to
Bo-
download?
both things are correct. So MSMS are available. We have published them before. So if we look in the papers that we have published in the last 20 years, you'll see that there's many MS spectra. The problem is that the first ones were not with the same quality and the same accuracy as the last ones. But also we have standards. So we have standards for all the urolithin A glycans and also some glucuronides and some sulfates. So it's feasible to work with them very nicely.
Would that be something that, because accessing MSMS spectra from literature is quite difficult, would that be, getting the raw data or at least like something that is
Yeah,
uploadable or even downloadable available to the
yeah,
community?
yeah. I think that some of the papers they have been deposited in metabolites, in the database metabolites.
Oh,
So there's
perfect.
a lot of information about that. But yeah, I think if somebody's interested in this, we could give the information on the spectra that we have in our lab as well. I'm sure that Carlos
Wonderful. That's great.
Carlos has been working in my group. He's a postdoc and he's been is of course willing to help anybody that is interested in the field and looking at the mass spectra and the MSMS or whatever.
Yeah, that sounds like a great idea, and we'll definitely reach out to Carlos and to you to get that, happening for everybody. Going on to the next question, what kind of methodology do you use to identify urolithin A? You hadn't highlighted the columns that you used or, the methods that were used to identify these metabolites.
Well, first, when we discovered the urolithin A and other urolithins, what we did was to isolate the molecule and then have first the MS, and then finally we had to do some NMR. But we need to isolate enough amount in order to have this spectra. And after that, we synthesized the molecules as well. So we have synthesized the different compounds. Well, sometimes when we have a new metabolite, well, I'm glad to say that for people that has been educated in the old analytical chemistry, and we've been using the UV spectra as something that is very useful, and not very sensitive, but is very useful because you can have a nice picture of the UV spectra, and this is very different from one phenolic compound to another. And then you can differentiate very easily with the UV spectrum. So we are able to see when you have a UPLC with a very sensitive UV spectrum to discover new spectra, UV spectra of compounds. And even if they have the same mass, we can have somehow a clue that this is a different molecule, and then we can isolate this metabolite and then run the NMR or other additional activity or the additional methodologies to identify. But again, this is something, if somebody has problems with this, we will be more than happy to help anyone that is interested.
Since you had said that the main antioxidant isn't
Punicalagin.
Punicalagin, that's a hard word to say.
Yeah.
And it's actually urothilins. Oh, I'm gonna mess up all these names. What concentration levels are they detectable in plasma? and any special treatment for their detection. Maybe,
Yeah.
I mean, you did mention that you're concentrating the samples, but
Yeah. Well, I
anything
can.
that you'd like to speak about their levels?
Well, I can tell you that normally we try to use the biological fluids, plasma, and urine without concentration. This is what we try to do because we don't want to modify this very much. So we prefer to work with the natural material and just with the addition of some solvents in order to stabilize the sample and filter and prepare the sample. But the detection limit of these compounds in plasma and in urine is very low. We have detected concentrations below nanomolar in plasma and in urine, of course. But the problem is that sometimes you can detect at lower, even lower concentrations, but not quantify. So I think is that you can concentrate if you want, but in our case with direct injection is perfect. We don't need really to concentrate. Sometimes when we don't see something with the UV detector, then we go to the cutoff, and then we look for the masses that we know that they produce and the MSMS, and then we can detect them and quantify them because we have the standards.
Great. And I think maybe many of us are thinking about how much pomegranate juice do we actually have to drink. Regarding the consumption of the juice itself, how much pomegranate would it take to result elevated levels of urethalins in the blood?
Yeah, probably if you drink 50 milliliters, you will have good levels of urolithins in your fluids. But if you eat walnuts are very rich in elagitannins as well. If you eat 30 grams of walnuts, then you have a large amount of urolithins. So it's because the number of the quantity of elagitannins in the walnut is higher than in the pomegranate. Not higher, is because punicalagin it only gives one molecule of punicalagin gives one ellagic acid, but when you look at the walnut, you can have many more ellagic acid units after one single molecule of elagitannin. So it's really interesting to say that with a very small amount, you are able to see urolithins. I can tell you as well that with tea, if you drink tea, the concentration of elagitannins there is very low, and you are able to see urolithins, and you are able to detect the different metabotypes after the intake of tea. So it's not. I think it's not a real problem. The detection of these metabolites. They're very easy.
Yeah, it sounds very easy to show up in the blood, as you said, 50 mils of pomegranate juice. Moving to the next set of questions, these are specifically about urolithins. You had stated that isourolithin A and urolithin B circulate in plasma, but before you stated that the main products are glucuronides and sulfates. Do we have data on concentration levels?
Yeah, the same kind of concentration that I mentioned before for glucuronides. What you see in plasma is not the glycons, it's just the glucuronides and the sulfates. And you can find them at concentrations in very low nanomolar concentrations. Up to that, you can see them. But normally, in some cases, you can reach as high as 1 micromolar or 2 micromolar. In the micromolar range, you can also see concentrations in plasma and urine. The problem with the plasma is that you you have a snapshot of what is circulating in plasma when you take a blood sample. And then unless you do a pharmacokinetic study in which you see, and in this case it's not pharmacokinetic because it's just you are eating the elagitannin then in the gut is producing the urolithins, and the urolithins are then absorbed and circulate in plasma. So probably you will see urolithins in plasma probably 2 or 3 days, up to 2, 3 or 3 days after the intake of the food containing the L-tannins. So this is something that has to be considered, but the concentrations can be as high as several micromolar.
Okay. To add to that, have you looked at the bioactivity of phase 2 metabolites of the different urolithins? You had shown a figure that had more than just A and B. There were several urolithin isomers, but several that you have found in red and blue and things like that.
Yeah. Yes, well, the phase 2 metabolites are the ones that circulate, and unfortunately they have less activity than the A-glycans, the urolithins that you can synthesize in the colon. Then the question is if these glucuronides are somehow released. Well, they release probably the A-glycan under some inflammation situations. And also what we know is that the sulfates of urolithins, they reach the brain. They reach the brain, and the A-glycan is also present in the brain. So this is that we have published recently in the Nature Communications paper. I think that probably the sulfates is going to be a very important group of metabolites, because they can be much easily broken down in tissues to release the A-glycan, which is the active molecule, rather than the glucuronides.
Regarding your findings about the different metabotypes in general, are you able to relate specific metabotypes to other known biomarkers?
We know that there are some. If we look at you mean biomarkers of other phytochemicals or biomarkers of health. What I can tell you that there is no correlation of, at least with other kind of biomarkers of phytochemical metabolism, like equol, for instance, or enterolactones or enterodiol from lignans. Normally, there's no correlation between the different metabotypes with other kind of distributions in other molecules. But I think this is not being studied so far enough, because it's not an easy strategy. It's not easy to perform this kind of study. So I think this is an open field, which is exciting. We know that there are differences between different individuals, but we need to know what happened with different phenolics, the different families of metabolites. Regarding the correlation between these metabotypes and biomarkers of health, I have no information about that. I know that the metabotype B, for instance, is more, is less healthy considering cardiovascular disease. But this is probably true because metabotype B is more prevalent in the elder people. So there's some change from metabotype A to metabotype B with age, and then this means that there's a change in the gut microbiota as well. And this is already being reported, that the microbiota changes with age. So this also is reflected with the metabotypes, but I cannot say anything really relevant about that apart from what I said.
Great, thank you. The last question on metabotypes, we have a few more questions left, but is regarding the mechanism of specific urolithins. What is the mechanisms of these urolithins, especially in metabotype Bs, and how it interacts with cholesterol metabolism, for example? Are there other in vitro studies that kind of can tell us more about this, for example, on hepatocytes?
Well, in hepatocytes, I cannot tell you because there's many studies already in vitro. But most of the studies are done with the A lichens, with the glucuronides and the sulfates that are the ones that reach the cells really in circulation. I think there's only very few of them, and they also show some anti-inflammatory effect. Regarding the cardiovascular, let's say cholesterol or lipids or whatever, I cannot say what's the effect. Probably it's an indirect effect through the gut microbiota and the interaction with other metabolites. But in the case of inflammation, it seems that these glucuronides of urolithin have been responsible for anti-inflammation effect in endothelial cells in vitro studies. And in this case, it was done by somebody in our group, which is Juan Jiménez Bastida, that was the first author of the study, working on looking at the anti-inflammatory effect of urolithin's glucuronides in these cells. And he found that there were some relevant effects, particularly in the inflammation induced by TNF-alpha, and this was relevant because these were the glucuronides, the ones that were doing some effects in vitro. In vivo is difficult to say, but we saw some effects on the clinical trial that we did with pomegranate, and we saw some effects, particularly in the urolithin A. It seems that urolithin A is more active in most of the studies than isourolithin A. So that is the main one in the main metabolite in metabotype B. But well, is something that is, of course, needs to be studied in more detail, but so far looks like a very promising group of molecules to study.
Yeah, they're definitely very interesting. And with any talk or lecture around food, I think a lot of us are genuinely interested in knowing, how much to eat or what to avoid. So there was a question about if there are any reported side effects on saffron and or in general urolithins when we consume things like pomegranate and walnuts, like you had mentioned.
Yeah, well, as far as I know, there is no report on the effect of polyphenols on side effects or negative effects on human health, and particularly this kind of elagitannins. Of course, it depends on the dose. If you eat too much, probably you can have some problems. But if you eat at the doses that are available in the food, then I think there is no risk for this kind of phytochemicals. The problem that happened a few years ago was with interaction with some drugs. There's some interaction with some drugs like statins, so there can be some interaction. There was a description of one study with one patient that have a rhabdomyolysis somehow enhanced by the consumption of elagitannins, but it was a single study, and it was not really very relevant. But if you eat the elagitanin-rich food at the normal doses, I think there's no risk at all.
Yeah, great. That's really good advice. And I think the last question, again, it's kind of this big overarching question is how do we maintain the healthy intestinal microbial colonies to have those strains? Because you did mention that it needs multiple different types of strains to have the effects that, we want to see. So how do we maintain a healthy gut microbiota is the big question, big takeaway.
Well, I can tell you with a healthy diet. With a healthy diet that is rich in plant-based food and very varied, that you have many different, phytochemicals and carbohydrates and complex carbohydrates in your diet, then you will be able to have a better gut microbiota. This is I think there's a close connection of what you eat and the phytochemicals that you eat and other components in the diet and the gut microbiota. And in this interaction with the gut microbiota, then there is the health effects. You know, I think this is the main advice to be given. If you want to keep a very healthy gut microbiota, you have to eat healthy food to give them the materials to grow better than other bacteria that they are not so healthy and that they can lead you to dysbiosis. That is what you don't want to have. So it's important to look after your gut microbiota by having very good food, rich in fruits, vegetables, and nuts and legumes, and fiber, which is good also for them. So, and then you will have a better gut microbiota.
Great. I think that is definitely the last question here. But we've also had a lot of great feedback from the participants or the attendees. They've shared that, you can find the MSM spectra on HMDB. Urolithin B is also available in NIS23. I will personally reach out to you about the spectra that, you had said that, we could share. But yeah, I think overall the talk was great, and I definitely learned a lot about pomegranate juice and the effects of those metabolites. If there aren't any other questions, I would like to open it up. Maybe Oliver, you have some comments or other questions that weren't addressed in the chat.
No, I mean, obviously, healthy food is difficult to define. Unhealthy food is relatively easy to define. So relatively means there's still arguments, of course, and this is a huge interest, at least in the United States, with all the food biomarker projects, but also in Europe, really. And so we, that is a little bit behind all these questions, and also the research that was presented by Dr. Tomás Barberán, to kind of understand how metabolites go into circular system and how they then affect or exert effects. And often enough, that is so far based on either in vitro cultures, then not really based on human experiments, and that is what Tomás Barberán also said. So, it's very hard to prove these things. And there's a lot of research that needs to be continued. We do know that microbiota is much more active than people thought maybe 20 years ago. And that they are being absorbed, so they are not just metabolized and stay there. So that means the analysis in blood and urine will remain relevant, so that we can distinguish what happens in the gut itself and what is being absorbed and observable in the human body, or the mouse for that matter.
For sure. Yes, I totally agree, Dr. Finn. I think it's a very relevant topic, and we start to know something about this, but we still have a lot of to be done. And I think that the nice equipment that we have now in hands in order to analyze with more sensitivity and more accuracy are essential in order to understand what's going on there. So, well, it's been very happy to be here with you discussing about these topics.
Okay, thank you so much for joining us at this evening for you. And, I wish a good day to everybody. And, for everybody who's still online, please join us back for next month's seminar. Arpana, do you do an interaction there?
I have some information there. So we'll have Dr. Kristen Overdahl, which was actually, she was supposed to present in January, and it's been rescheduled for next month. She'll be talking about metabolomics research at the NIEHS Intramural Research Program. So if you had registered for, her time in January and didn't get to, hear her, this is a good time to re-register and hear her talk in March. So with that, thank you everybody for joining, and thank you Professor Barberán for the great talk, and we'll stay connected. Yes.
Thank you. Good day.
Bye.
And
Thank you.
Bye-bye.